But there will be a Q&A time after that, and I wanted to clarify the ground rules for that by way of a personal story. 25 years ago, I was just entering my postgraduate studies at Cambridge, and like many American students, I was perhaps, overawed with the place, and perhaps a little overly earnest and eager to please. One day, there was a visiting professor who was an expert on the philosophy of Immanuel Kant, and he was lecturing. I'd had one undergraduate course on Kant's work, but had only really a cursory familiarity with it. And he was talking about some aspects of Kant's philosophy that I'd never encountered. And so I found myself asking a question, admitting my ignorance about this, and asking if he could give me a source whereby I could read more, since I did not actually know much about what he was discussing. There was this awkward pall that came over the room. He eventually obliged me and gave me a source, but afterwards my supervisor took me aside. He was very much a don of the old school, and Professor Michael Hoskin, a wonderful man, and he said in his high Oxbridge, if you'll indulge me, Meyer, he said, "I know in the States you've learned that the only stupid question is the one you didn't ask. It's different here." Then he went on to explain to me that in a competitive academic environment like Cambridge, it was very important not to ask questions that revealed ignorance. And he said, in future, if you have questions like that, come ask me privately, he said. I just want you to know that in this one particular respect, that is to say our Q&A time afterwards, it will be American rules. All questions are welcome. I know none will be stupid, obviously, but all questions are welcome from any point of view. And so with that, I'd like to kind of dive in on a very different way. In a very different way, I'm very happy to be in a British setting, in a UK setting. My thinking about intelligent design was actually, it actually germinated here in the UK when I was studying the history and philosophy of science. And in particular, when I was studying the scientific method for investigating the remote past, which Darwin himself pioneered. There's another reason, though, that I appreciate the opportunity to give this address here. The United Kingdom and Europe generally never had a Scopes trial. And I have found that lecturing in the United States on this topic, that the perception of what we are saying, the perception of our case for intelligent design, and its reception have been, I think, badly distorted by a fear of fundamentalism. Intelligent design has been equated in people's minds with young Earth creationism, no matter how clearly and patiently and sometimes emphatically we attempt to explain otherwise. And so it's nice to have a fresh start. Intelligent design is not a religious doctrine. It is not an interpretation of the Genesis text. It is, in fact, a scientific inference from biological evidence based on the same methods of reasoning that Darwin himself used in The Origin of Species. Now, the question of design goes very far back, as far back at least to the ancient Greeks. And the question of design, in essence, is the question of whether or not nature, particularly biological nature, owes its origin to purely undirected material processes, or whether mind might have played a role in what we see in the living world. Steve Fuller, the philosopher of science from Warwick University, has pointed out that this question of design has animated the whole tradition of British natural philosophy, of British science, going back to Boyle and Newton and to Faraday and Maxwell and to Darwin himself. And in the last couple of years, since the very big bicentennial celebration of the birth of Darwin and the 150-year anniversary of The Origin of Species, both of which occurred in 2009, there has been an extensive discussion worldwide about the legacy of Darwin. What did he establish? What did he leave us with? What were his key insights? And clearly, Darwin introduced, obviously, a theory, an evolutionary theory of biological change over time. He introduced to the biological establishment, the biological discipline, the concept of natural selection, along with Alfred Wallace. And thirdly, I would say, very importantly, he introduced a scientific method of investigating the history of life. Now, advocates of intelligent design dispute none of these things. We accept these things. For example, we accept that the process of natural selection is a real process. It produces real and demonstrable change in living organisms. Almost all of us accept that the universe and the Earth have a very ancient history. I myself think the Earth is billions of years old. These are simply not our animating concerns in the intelligent design research community. Instead, the concern that has been animating, that has been at the forefront of our minds, is the question of design, and in fact, the very question that many historians of ideas, scientific historians, and evolutionary biologists say is the central legacy of Darwin's theory, the central thing that he's left us with, which is the idea that natural selection acting on random variations, or we would now say mutations, has been wholly sufficient to produce everything we see in the living world, such that there's no evidence of actual design. Design is merely an appearance. So the central legacy, that as has been emphasized by many authors, is the denial of the design argument, the refutation of the classical design argument. Here's Francisco Ayala, and he puts it this way. He says, it was Darwin's greatest achievement to show that the directive organizations of living beings can be explained as the result of a natural process, natural selection, without any need to resort to a creator or any other external agent. Richard Dawkins, who's been very much in the news here lately, puts it this way in the first page of The Blind Watchmaker. And I know Dawkins is not regarded in some circles as the serious figure that he once was because of some of the media involvements, but we actually appreciate him very much because of the clarity with which he defines the issues. And I think the opening page of The Blind Watchmaker is one of those examples. He says that biology, and this is from a Darwinian point of view, he says biology is the study of complicated things that give the appearance of having been designed for a purpose. And of course, for Dawkins and other classical Darwinists and neo-Darwinists, the key word there is appearance. Living systems appear to be designed, but they're not really designed because a purely undirected process, namely natural selection, has produced that appearance without being directed or guided in any way. We have, as Ayala has also put it, design without a designer. We have the appearance of design, the illusion of design, without an actual designing mind behind it. Now that's the proposition that interests us. Is that true or false? The intelligent design idea is that there are certain features of living systems that point to an actual designing intelligence, not merely to a designer substitute, an undirected mechanism that is mimicking the powers of a designing intelligence. And that's what we're going to get into. Now I think before we do, though, it's important to briefly review the logic of the Darwinian denial of design, because it is built into the structure of The Origin. Chapter 4 on natural selection is crucial here. Darwin uses a number of examples, and his starting point is his observation in the 19th century of the power of artificial selection, of breeding experiments. These were well known at the time, and they provided for many biologists prior to Darwin a compelling argument for design. In the 19th century, the adaptation of organisms to their environment seemed to be an overwhelming argument for the idea that there was some kind of design. Fish swim in the water, they have gills and fins. Birds fly, they have wings, certain kind of lungs that allow them to process oxygen in order to sustain flight. And my example is of lowly sheep. If you imagine that you're a sheep herder in the north of Scotland and you want to breed a woollier breed of sheep, what do? Well, in the 19th century, it was obvious. We know it today. It's artificial selection, artificial breeding. You choose the woolliest males and the woolliest ewes, and you allow only them to breed. And you do that over a series of generations, and after a while, you will have a woollier breed of sheep. Now, Darwin came along, and he didn't use the example of sheep, but he used other similar examples. And he said, what if, in effect, adapting his examples to mine, he says, what if you have, instead of the artificial or intentional selection of the breeder determining the features to be amplified, what if instead you have, for example, using my, a series of very cold winters, such that after a while only the woolliest sheep have survived and then you do that generation after generation after generation. There is a series of very cold winters. Will not the effect of that natural variation in climate be exactly the same as what the intelligent agent was able to do quite intentionally? And of course, Darwin answers that rhetorical question in the affirmative. Nature can do what the intelligent breeder can do. Nature can produce adaptation and produce the effect that heretofore we thought only intelligent agents can do. And for that reason, Darwin is very clear that the mechanism of natural selection eliminates the need to invoke a guiding intelligence of any kind. In fact, later, after the publication of The Origin, he clarifies this in a series of letters to different scientific luminaries in the day. Here's one. He says, "There is no more design in the variability of organic beings and in the action of natural selection than in the course in which the wind blows. It's apparent design explained by an undirected process that mimics the powers of a designing intelligence, but which is not itself intelligent in any way. And therefore, the most difficult things in living systems can be explained without design, thus the claim that Darwin has refuted the design argument." Now, bringing this up to date, it's important to ask a few questions. The first is adaptation, the effect that most impressed 19th century biologists of actual design, is adaptation the only appearance of design in biology? And if not, has natural selection or some other similarly undirected material process explained all these other appearances of design? Today, you may know there is a very spirited scientific discussion going on about the adequacy of natural selection and random mutation to produce not the minor variations that were used in my illustration, woolly or slightly less woolly sheep, but rather the fundamental innovations in the history of life, the origin of animal body plans in the first place, for example. And there are many evolutionary biologists who are now saying that the Darwinian mechanism of mutation and selection, the neo-Darwinian mechanism, is insufficient to produce the large-scale innovations in the history of life as opposed to the smaller-scale variations. There's a group, in fact, called the Altenberg 16, an international group of evolutionary biologists who are now calling for a new theory of evolution because they think the Darwinian mechanism is insufficient to produce that fundamental innovation. Now, I merely flag that issue tonight. I'm not going to join that argument one way or another in this talk because my story is a little different. My interest was not in the sufficiency of the specifically biological form of evolution that Darwin championed, but I got interested in some ways, a more fundamental question, which is the origin of life itself. People may, in the audience, I'm sure, be familiar with a distinction in evolutionary theory, a distinction between biological evolution, which attempts to account for new living forms from simpler pre-existing forms, and chemical evolution, which attempts to account for the origin of life in the first place from simpler pre-existing chemicals. We can contextualize that distinction by looking at Darwin's tree, the famous depiction of the history of life, where the branches represent the diversification of new living forms, and the trunk of the tree represents the very first form of life. Conceding for the sake of argument the sufficiency of modern neo-Darwinism, that still leaves open a very important question. Perhaps if it's true that Darwin accounted for all appearances of a design that arose from the beginning of life forward, is it the case that there is still no evidence of design at the point of the origin of the first life? We now think of the origin of the first life as the origin of the first cell. Now, as I got into the topic of origin of life studies in my postgraduate work, this was what animated me. First of all, it was clear Darwin had not explained the origin of the first life. It was something that he did not attempt to do. It's no fault of his. It wasn't the purpose of the origin of species. He started by presuming, as he put it, one or very few simple forms. And of course, this was something that puzzled me. The consensus of intellectual historians of science, of evolutionary biologists, was that Darwin had refuted the design argument. And yet there was a very important event in the history of life involving the origin of life itself that everyone also acknowledged had not been explained or explained away by Darwin's theory or, for the most part, any other undirected materialistic process. And so I began to wonder, well, why is that? Why was that? In the 19th century, scientists were well aware that they did not have a theory of the origin of life. Darwin wrote one letter about the topic to a friend, Joseph Hooker, in which he speculated that perhaps life might have existed in a warm little pond, but he was very, very forthright to confess his ignorance of the whole topic, of what might have happened. So there was a kind of historical puzzle here for me. On the one hand, you have people saying, historians of science, intellectual historians, evolutionary biologists, that all evidences of design have been explained away, that the design argument was refuted by Darwin. But on the other, we have at least one possible locus of design, which Darwin's theory admittedly doesn't touch. Now, why the confidence in the refutation of design and yet the stated agnosticism about, or the stated understanding of the limits of Darwin's theory? Well, the resolution to that historical puzzle is that in the 19th century, evolutionary scientists who followed in the wake of Darwin were convinced that life was exceedingly simple in its cellular form. The simplest form of life was exceedingly simple in its structure. I love these quotations from Thomas Henry Huxley, Ernst Haeckel. They were known for their advocacy of something called the protoplasmic view of life. It was the idea that, in Haeckel's words, the cell is a simple, homogeneous globule of plasm. We have something in the United States called Jell-O that's served at holidays. And this was kind of the idea, this gooey stuff, undifferentiated, no interesting structure. And the idea was that because life was so simple, it was easy to imagine, even if we didn't know how, that a couple of simple chemical transformations could produce this protoplasmic material, this essence of life, without any guidance or direction. And both Haeckel and Huxley came up with theories of this one or two step origin of life by reference to simple chemical transformations, starting from very simple chemicals. Now, these weren't developed in any detail, but because people assumed that life was so simple, it was also thought that it was unnecessary to do much more than they had done. And so, conjoining their ideas about the origin of the first life with Darwin's idea about what happened after that, it was possible to paint a kind of seamless materialistic account that eliminated, again, any need to talk about design. Now, obviously, our view of life has changed dramatically since the 1860s and 70s. In the 1890s, scientists began to learn about metabolism and about these large molecular weight molecules called proteins. And in the 1920s, there were new theories of the origin of life that were advanced to take advantage of, or to try to account for that increasing view of the complexity of life. But the really dramatic change came in the 1950s as scientists began to elucidate both the structure of proteins and the structure and function of DNA. And of course, in my book, I highlight in particular the importance of the discovery of Watson and Crick, in which they were able to elucidate in 1953 the chemical structure of the DNA molecule. But I also emphasize something which I think is even more fundamental, at least with respect to the question that interests me, which is the question of the origin of life. And that is, I think, the very brilliant insight of Francis Crick. It was termed a conjecture or a hypothesis. It came 4 years after the big and famous discovery. It was called the sequence hypothesis. And according to Crick, the four chemicals that run along the interior of the spine of the DNA molecule, called nucleotide bases, that these four bases function like alphabetic characters in a written language or digital characters in a section of computer or machine code. That is to say that the function of the bases in the DNA molecule is not determined so much by their shape or by their chemical nature, but instead by their specific sequencing, by their arrangement. It's the arrangement of the bases, which we represent with the letters A, C, G, and T, that allows them to perform their most crucial function, which Crick already was beginning to suspect had something to do with the construction of proteins. In fact, what he was entertaining was the idea that the arrangement of the bases conveyed instructions or information for directing the construction of protein synthesis, or synthesizing proteins. Now, to appreciate this, we need to understand just a little bit more about proteins. And I know some in the audience are scientists, others aren't, so forgive me for trying to hit the target right in between. What we have are we have the chemical formulas for some the subunits out of which proteins are made. These are called amino acids. And I have a simple visual aid to get this across. These are called snap-lock blocks. I stole them from my kids when they were little. It said on the box, ages 2 to 4. So I thought this would be a fitting scientific illustration. I'm using each of these snap-lock beads to represent an amino acid. Proteins are long chain-like molecules that fold into specific three-dimensional shapes. They're formed from smaller subunits, smaller chemicals called amino acids. There are 20 different amino acids that can be part of proteins. The protein-forming group of amino acids comprise 20 varieties. Now, depending on how these are arranged, the molecule, the chain, will fold in different ways. And if it folds in just the right way, it will have a three-dimensional shape that will allow it to perform a specific function inside the cell. Proteins are like the toolbox in the cell. There are lots of different tools, lots of different proteins, and each one of the protein tools performs a job in virtue of the specific three-dimensional shape it possesses. Think of a saw or a hammer or a spanner. Each one of those has a specific shape, and the job that it can do is related to the shape and form that it possesses. It's the same thing with proteins. So we have, for example, these are some pictures of different protein motifs. You can find these on the internet. It's fascinating what we know about the structure of proteins. The first three-dimensional structure was elucidated in 1958 by Kendrew in Cambridge. But now we've had thousands of proteins that have known structures. And each one, again, one of those structures is relevant to performing a specific task in virtue of its three-dimensional conformation or shape. Here's an example of an enzyme that breaks apart a two-part sugar, a disaccharide. The sugar is a little barbell-shaped thing. It nestles into those grooves, and as a result of that, the enzyme breaks that apart. It catalyzes a reaction that would not occur nearly as frequently without the enzyme. And so proteins do lots of important jobs in the cell. They catalyze reactions. They form the structural parts of molecular machines. They help to process and copy genetic information. They're really the workhorses of the cell. Now, three key features then of proteins. They have shapes that perform functions. Those shapes are produced by folded chains, and the precise sequencing of amino acids determines the folding and then the function of the protein. Now that leads to a really interesting insight. And again, Francis Crick was at the forefront of this. Proteins they manifest or display a property, an information theoretic property that we could call sequence specificity. Something is sequence specific if the function of the whole is determined by the arrangement of the parts. Words are sequence specific. Sections of computer code are sequence specific. And proteins are sequence specific. In fact, Crick likened the sequence specificity of proteins to the old typefaces on the little blocks that were used to make headlines back in the 1950s. You can see the chemical structure of the amino acids. There's a certain part of it, the backbone is repeating. It's the same chemical structure. And the little sticky outies, also known as side chains, are different in each case. They're kind of like the font faces on those blocks, those type blocks. This is Crick's own analogy. And what he was trying to emphasize was that the function of the protein is determined by the arrangement of those side chains, the arrangements of, in a sense, the fonts, by analogy. Again, it's this specificity of arrangement that is critical to biological function. Now, his sequence hypothesis took this insight even deeper, because what he suspected was that that the sequence specificity of the proteins, of the amino acids in the proteins, derived from a prior sequence specificity, from the specificity of arrangement of the bases, the information along the spine of the DNA molecule. And That was his essential insight. It's, by the way, if you know anything about Crick's career, it's very interesting. He did not have a PhD in biology when he made the great breakthrough with Watson. He was working on a PhD in physics at the time, but he was also, during the war, a code breaker, and I think that's somewhat apropos. In any case, his idea of the sequence hypothesis was eventually confirmed, not by a single experiment, but by a whole flurry of experiments on both sides of the Atlantic over about a 7 to 8 year period. And as a result of that, scientists have been able to characterize what's sometimes called the gene expression system, or what I like to think of as how the information in DNA generates a protein, sometimes called protein synthesis. We have these required courses in American education called civics, and there you have to learn about how a bill becomes a law. Well, this is how a gene produces a protein. And I have a little animation to make this clear, because one picture here is worth a whole lot of my words. This is an animation of how the digital information in the DNA directs the construction of proteins.
In 1957, Francis Crick first proposed that chemicals called bases along the spine of the DNA molecule function as alphabetic characters in a written language or digital characters in a machine code. This animation shows how this digital information directs protein synthesis. First, a large protein complex separates the tightly wound strands of the DNA to prepare it to be copied. During this process of transcription, a protein complex called a polymerase produces a single-stranded copy of the original instructions. Here we see this copy, a messenger RNA molecule, being constructed inside the polymerase as individual bases are added to the growing strand. Now we see the polymerase in action from the outside as it spits out the messenger RNA transcript. Next, this RNA transcript approaches and passes through a molecular machine called the nuclear pore complex, an information recognition device that controls the flow of information in and out of the cell's nucleus. Now we see the genetic assembly instructions on the messenger RNA approaching and arriving at a two-part chemical factory called a ribosome, the site of protein synthesis. As the messenger RNA transcript passes through the ribosome, the process of translation begins. During translation, a mechanical assembly line builds a specifically sequenced chain of amino acids in accord with the instructions on the transcript. These amino acids are transported from other parts of the cell by molecules called transfer RNAs, which link specific sequences of bases to corresponding amino acids. The sequential arrangement of the amino acids determines the type of protein constructed. When the construction of the chain is complete, it is often transported to a barrel-shaped machine that helps fold it into the precise shape required to perform its function. After the chain is folded into a protein, it is released into the outer cytoplasm to do its job in the cell.
So this is what's become of Huxley's undifferentiated protoplasm. The simple cell is no longer simple. Now, one other analogy that may help understand what's going on here. I'm from Seattle, where we have the people from Microsoft and also the Boeing people who build airplanes. And there is a technology, a human technology today called CAD/CAM, Computer Assisted Design and Manufacture. It occurs as engineers sit at a console, use a computer to design mechanical parts. They make selections of parameters. Those selections are then encoded digitally. They're transferred down a wire. They're translated into another machine code that can be read by an assembly apparatus, and then that assembly apparatus takes those instructions and, for example, might use them to put rivets at a particular place on a wing of an airplane or something. It's a very common technology in which digital information is being used to direct the construction of mechanical parts. This is what's going on inside living systems. This has been part of the mind-blowing development that's called the molecular biological revolution. It has raised some deep and profound questions in another area of science. And I call this, the questions surrounding this, the DNA enigma. The DNA enigma is not the structure of the DNA molecule. Watson and Crick did a wonderful job of elucidating that. The DNA molecule is not the mystery of where biological information resides. We know where at least a lot of the biological information resides, and that is in DNA and RNA. There are other sources of information in the cell that we now know about as well, but we'll set that to the side for now. So the DNA enigma is not where the information resides. It's not the structure of DNA. The DNA enigma is not even what the information does. The animation just showed, in sketch at least, what the information in DNA does and how it directs protein synthesis. The DNA enigma instead concerns the question of origins. The DNA enigma is about the origin of information, and this mystery of the origin of this information. Where did it come from? How did the DNA molecule acquire this precise sequencing that allows it to direct these mechanical operations? That's the key question, and it is closely related to and at the heart of another question in science, which is the question of the origin of life itself. German biochemist Bernd Olaf Küppers puts it this way. He says the problem of the origin of life is now clearly, basically equivalent to the problem of the origin of biological information. Think of it this way. I used to ask my students this. They would understand these things better than people over 30, I suppose. But I used to ask them about their computers and say, if you want to give your computer a new function, what do you have to give it? They would immediately say code, software, a program. Well, it turns out the same thing is true in life. If you want to build a new form of life from a simpler pre-existing form, you have to have new forms of information. If you want to build life in the first place from simpler chemicals, the chemicals have to be arranged in precise ways that convey information. In particular, convey information for building proteins. And so, as we begin to think about the origin of life, to make that problem at all tractable, we need to begin to think about where did the information come from. Now, I want to offer just a word of clarification about the kind of information that we're talking about in biology, because there are two distinct concepts of information that could be relevant in this context. One is the purely mathematical notion of information. This is the concept of information that was put forward by Claude Shannon, the famous information theorist in the late 1940s. And what Shannon did was link the notion of information to the idea of the reduction of uncertainty. Very intuitive idea. Take, for example, a coin. If I toss a coin and it comes up heads, as soon as it comes up heads, my uncertainty has been reduced about the outcome by half. There were two possibilities beforehand. The coin came up heads, and now I am able to eliminate my uncertainty about whether it would be heads or tails. Shannon linked this notion of the reduction of uncertainty also to probability, because there is a 1 in 2 chance, a 50% chance of it being either heads or tails, and once the coin comes up heads, it has eliminated the other half of that uncertainty, if you will. So the more uncertain an event, the more improbable it is, and the more information is conveyed when that event occurs. Take, for example, the rolling of a die. If I roll a die and it comes up a 2, now I've eliminated more uncertainty because the probability of it coming up 2 was 1 in 6 instead of 1 in 2. And so what Shannon did was relate the concepts of information, reduction of uncertainty, and improbability. The more improbable an event, the more information it conveys in this strictly mathematical sense. But Shannon and one of his collaborators, Weaver, were very clear to point out that their form of information had nothing to do with meaning or function. The sequence on the top in the slide behind me conveys a calculable amount of Shannon information. The amount that is inversely related to the probability of that particular sequence occurring given the keyboard that we have. The sequence on the bottom conveys an almost identical amount of Shannon information, but clearly it also manifests another property that is not captured by the merely mathematical analysis of probabilities, of Shannon's theory. We call this property specificity. On the lower string, it's clear that the arrangement of the characters is crucial to the function that the characters perform. Now, the term of art in origin of life studies, this term came from the German origin of life scientist Leslie Orgel, is the idea of specified complexity. Shannon information, which is a measure of improbability, is also sometimes equated with the concept of complexity. If something's highly improbable, it's also complex. What Orgel said was that in biological systems, like in language and computer code, you have complexity, a lot of non-repeating, highly improbable arrangements of characters, but those arrangements are also specified to perform a function. Now, Crick was very aware of this distinction. Francis Crick was very aware of the distinction between the merely mathematical conception of information and the more, in a sense, common conception that conveys this idea of specificity. And he was insistent that when we're talking about the information in living systems, we're not talking about mere Shannon information. In his own words, by information, I mean the specification of the amino acid sequence in the protein, or, he says, information means here the precise determination of sequence, either of bases in the nucleic acid, in the DNA, or the specification, the precise determination of sequence in the amino acid residues in the protein. So I've used a few terms here: sequence specificity, specified complexity, and information. The kind of information we're talking about involves sequence specificity. It's specified and complex, not merely complex or not merely improbable. Therefore, it's not just Shannon information. And that is the key thing that has to be explained to explain the origin of life. Now, interestingly, Richard Dawkins, who brings such clarity to the debate, has also commented on this. He has pointed out that the machine code of the genes, the information in DNA, is uncannily computer-like. Apart from differences in jargon, the pages of a molecular biology journal might be interchanged with those of a computer engineering journal. In other words, what we have here in living systems is a strike at the very foundation of life. In the very simplest forms of life, we have very striking appearances of design. Our own local hero in Redmond, Washington, where I hail from, Bill Gates, puts it this way. He says, the DNA is like a computer code, only much more complex than any we've been able to devise. We have a striking appearance of design. Now, the question that animated me, that seized my imagination and interest as a postgraduate student in Cambridge, was this question of the origin of information. My own story is that I came out of a field of physics, geophysics, and I was doing digital signal processing as part of my job. I worked on seismic signals and traces. And I attended a conference that came to the city where I was working on the origin of life. Now, I was under the impression at that time, I was a young scientist, that the evolutionary biologists had that question and pretty much everything all sewn up. There were many very prominent origin of life biochemists at this conference, and I was shocked to find out that the leading people in this field were very candid about their, in a sense, ignorance, their lack of an adequate evolutionary explanation for the origin of the first life. In fact, at that conference, the key thing that they kept discussing was this problem of the origin of genetic information. Where did the information come from? And so as I entered my postgraduate studies a year or so later, I naturally became fascinated with this, and I eventually chose the topic of the origin of life and the related questions about scientific methodology as my PhD topic. And as I got into this, I came across something that was very helpful for framing the issue. It was a famous book, small book, by Jacques Monod. He was a French molecular biologist. He was a close colleague of Crick's, especially in the 60s. And he simply argued that if we were to approach any topic scientifically, we needed to invoke certain basic kinds of explanatory categories. We needed to either explain events by reference to chance, stochastic or random processes, or by reference to necessity, law-like processes. If I drop a ball, or if I let go of a ball and it falls to the earth, we say it falls because of a law-like necessity. It falls by the law of gravity, by necessity. So Monod's point was, when we're explaining things in science, we want to explain things by reference to either chance or necessity, or the combination of the two. And as I began to investigate the different theories that have been proposed to explain the origin of life, and in particular the origin of the information necessary to produce it, I found that they broke nicely into these three categories. The first approach early on was the approach called chance. It just was the idea that some kind of random association of these chemical building blocks that occurred on the early Earth in something called the prebiotic soup, and though it may not have been that probable, eventually the odds were in its favor, and these information-rich molecules would have arisen. There was a famous scientist, George Wald, who put this idea forward in 1954. Now, by 1971, it's very striking as you read the literature in Origin of Life biology. Most of the leading researchers had utterly rejected the pure chance hypothesis. Here's one, A.G. Karen Smith, a Scottish chemist, who says blind chance is very limited. Low levels of cooperation he can produce exceedingly easily, the equivalent of letters and small words. But chance becomes very quickly incompetent as the amount of organization increases. I've got a visual aid. Again, I had to look this up online last night to find out if you all play Scrabble as we do. But there is a British Scrabble association, so I'm sure you're familiar with this game with the letters. And I used to do a kind of thought experiment with my students to convey to them why the Origin of Life scientists had given up on chance. What I would do is I'd walk around the room with this bag of Scrabble letters, have the students choose a letter at random, and go up to the board and write whatever letter it was. And almost invariably, within 3 or 4 letters, the board would be full of gibberish. Now, on one or two occasions, the students would produce a few small, not so much words, or maybe word-like syllables or something like got or bit or something. And students being students, they would start to kind of do cat calls and say, yeah, we're showing you up. But I always won the experiment, and the way I always won is what Karen Smith's talking about. I just let the students continue to select letters, because very soon the gibberish would dwarf whatever little tiny elements of possible meaning were there. And this is Karen Smith's point. If you want chance to produce a little bit of information, it may work out for you. But as the amount of information required increases, unless you have vast probabilistic resources, lots and lots of trials, you can't get the job done. Now, one of the things that contributed to this view among Origin of Life scientists was something called the combinatorial problem. There's a way of illustrating that with a simple device, a bike lock. If you think of the number of different ways of arranging the dials here, you realize that each one of these dials on the bike lock has 10 possible settings. And there are 4 dials. So the total number of possible settings is not 10 plus 10 plus 10 plus 10, but rather 10 times 10 times 10 times 10, or 10,000 possible combinations. Now shift the context a little bit. I had my graphic artist design this. I don't think there are locks like this, but given the crime rate these days, maybe it would be good to have one. This is a bike lock with 10 dials. Now, again, the number of possible combinations and the odds, or probability rather, of finding the right combination by chance increases precipitously, exponentially. Now we have 10 times 10, 10 to the 10th power, 10 billion possible combinations. Now imagine you're a thief out in front of the hotel wanting to pick a lock like this. If you have just 30 seconds or a minute before the porters come out, is it more likely than not or less likely than not that you're going to find that correct combination by chance? Well, obviously, it's vastly more unlikely that you will find that than it is likely that you will find it. And now, the same thing ends up applying to the problem of generating even a single information-rich DNA or protein molecule by chance. I'll illustrate the principle with proteins. If you take just a short little section of a polypeptide, a short little section of amino acids with 10 sites, given that you have 20 possible amino acids that could attach in this growing chain at each site, you have 20 times 20 times 20 to the 10th power possibilities, which works out to 10 trillion possible combinations for a very short little section of polypeptide. Now, most proteins are many hundreds of amino acids long. The average protein is something around 300 amino acids. The protein, I have a colleague, Doug Axe, who's here with us tonight, who's done experimental work on very modest length proteins of a shorter length, about 150 amino acids. And if you begin to think about this mathematically, a very obvious problem arises for the chance hypothesis. If you have 150 sites with 20 possibilities at each site, you have 20 to the 150th power possibilities, which in base 10 computes to 10 to the 195th possibilities. So the odds of getting the probability of getting any particular sequence stands at 1 chance in 10 to the 195th. Now, we happen to know that there's some variability admitted at these amino acid sites. So the problem is not quite that severe. What Dr. Axe has done is investigate a very important question that arose in the 1960s as scientists were beginning to think about the probability of chance alone generating these large information-rich biomolecules. And the question was this: how rare or common are the functional sequences, functional proteins or information-rich DNA molecules, among all the possible combinations? If that group of 10 to the 195th is populated liberally with functional combinations such that almost anything you try will give you a combination that will, by analogy, unlock the function or perform a biological function, then chance alone might get the job done. But what Dr. Axe found was that the space of possible arrangements of amino acids, what mathematicians call a combinatorial space, was very, extremely, I can't use enough adjectives, infrequently populated by functional combinations. He did some very careful sampling work called site-directed mutagenesis and showed that in the case of a very simple 150 amino acid long protein, the odds of getting correct sequencing were about 1 chance in 10 to the 77th power. Now, I won't go through all the math here. I do it in the book. But when you combine that insight with a number of the other factors that are relevant to computing the probability of generating a single protein by chance, including the important factor of the number of trials that you might have since the very beginning of the universe, you come to a startling result, and that result is this: that if every event from the beginning of the universe till now was devoted to searching for one of these functional combinations among all the possibilities, which is, again, a kind of an absurdly generous assumption, you would not have time to search more than about a trillionth of all the possibilities. A little tiny needle in a vast haystack the size of a galaxy. And so, as scientists began to grapple with this, to understand that it was vastly more likely that chance was false than it was true, that chance could get the job done rather than that it couldn't get the job done, they began to reject it and look for other approaches to the origin of information and the origin of life, as it related to the origin of life. Now, obviously, the Darwinian approach in biological evolution has never been to rely on chance alone. That would be a kind of caricature of the theory, one which some people promulgate, but I'm not doing here tonight. Instead, the Darwinian approach has always been to conjoin random variations or random mutations, chance events, with the law-like properties of the process known as natural selection. And so it was natural that scientists attempting to explain the origin of life would early on invoke selection, natural selection, to operate in conjunction with chance as a way of trying to overcome these large probabilistic hurdles to the origin of information. Unfortunately, there was a real problem with this approach, and it was a logical problem that became apparent fairly early on. It's simply the logical fallacy of begging the question, and here's the problem. Natural selection is a process that involves differential reproduction. It is only operative or relevant once you have an organism that can copy itself and leave offspring of different varieties. So natural selection presupposes self-replication, the ability of an organism to copy itself. It presupposes a form of life, in fact. But what are we trying to explain? Well, we're trying to explain the origin of the information necessary to produce life, and self-replication in all living organisms depends upon sequence-specific, that is, information-rich DNA and proteins. In other words, we are invoking the very thing we're trying to explain in order to explain it. One scientist, the Nobel Prize-winning biochemist who works a lot on the origin of life, Christian de Duve, puts it this way. He says, theories of prebiotic natural selection are problematic because, quote, they need information, which implies they have to presuppose what is to be explained in the first place. I have a little illustration of the question-begging nature of this that I used to use with my students. I would depict an absent-minded philosopher of science walking home from class. This poor hapless soul, not paying attention to where he was going, would fall into a big pit. The pit was 30 feet deep. He found there was no way out. But then he began to reflect, to think deep philosophical thoughts, and he thought, you know, I could get out of here if I only had a ladder. And so the philosopher of science climbed out of the hole, went home and got a ladder, and jumped back in the hole and climbed his way out. This is logically illustrative of the problem of prebiotic natural selection. Now, it begs the question. Now, there's a more sophisticated version of this idea that is currently considered to be the most cutting-edge idea about the origin of life. It's called the RNA world. And the idea of the RNA world is that in the beginning was not a simple cell, but before that, there was a simple self-replicating molecule. And that once that molecule began to copy itself, it produced an offspring, a crop of offspring which could compete, and that's how the evolutionary process got going, which then in turn generated additional complexity. Well, there are a number of problems with the RNA world, and I'm happy to speak to them all in the question and answer time. I have an extensive chapter about this in the book. It's interesting to note, for example, that Christian de Duve and other people who are very much in the forefront of origin of life studies are now saying that we now need a pre-RNA world because the RNA world is so fraught with difficulty. But for the purposes of my talk, I want to just highlight one of those difficulties, and it is again an information problem. The RNA world envisions an RNA molecule capable of copying itself. The backstory on that is that they're called ribozyme engineers. Scientists are working very hard to try to get RNA to do the things that we hope it would do if the RNA world hypothesis were to be true. Have found that in the best of cases, they've been able to get an RNA molecule that can copy about 1/10 of itself, about 1/10 of the bases. But to do that, even to get that limited replicase capacity, requires a huge amount of information in the form of very precisely sequenced bases that have been sequenced by an intelligent agent, namely the ribozyme engineer. So what exactly does that simulate? Well, some might say it simulates the role of intelligent design, but setting that point aside, it certainly shows that the RNA world does not account for the origin of information. Again, it too presupposes information just to get that primitive molecular replicator going. So this whole approach of relying on natural selection, whether with simple organisms or simpler molecules, has run into a very imposing obstacle. Now, the third approach recommended by Monod's categories is the approach of necessity or law, or what goes by the name in origin of life studies as self-organizational scenarios. The idea here was very simple. It was proposed first in the late 60s and early 70s, and it was the idea that perhaps these large biomolecules could be likened to something like a crystal, where the forces of attraction between the constituent parts determine the order, the sequencing of the chemical constituents of the molecule. A crystal of salt has two ions, the sodium with the plus charge and the Cl with the minus. There's a force of attraction. That force of attraction generates, in a very self-organizational way, a beautiful, orderly crystal lattice. two scientists, Kenman and Steinman, proposed that perhaps that's what's going on to explain the origin of the information, the sequence specificity in proteins. Maybe that's what's at work in nucleic acids as well. Now, they after several years working on this proposal, they wrote a very important book called Biochemical Predestination. They weren't Calvinist theologians, they were chemists, saying that the structure of DNA, the information it contained, was predetermined or biochemically predestined by these forces of attraction between its constituent parts. Now, they ended up repudiating their own theory. They had invoked this initially to account for the sequential arrangement of the amino acids in proteins. They found that there were some slight differences in the bonding affinities between the amino acids, but none of them correlated to the arrangement of amino acids in known proteins. But with the DNA, they found that there was an even deeper problem. It was Kenyon who overtly repudiated this idea. And the problem was this, and I need to direct your attention to, good, it works. The structural formula for the DNA molecule. The pentagons in the sugars form what's called the backbone. And there's one on each side. The A's, C's, T's, and G's in these little boxes represent the nucleotide bases. Remember that it's the sequential arrangement of those bases that conveys the assembly instructions for building the proteins, for arranging the amino acids in the right way. Now, what they were trying to do was to explain the sequencing, why the arrangements are just the way they are, by reference to forces of chemical attraction. Now, notice that in the molecule, there are these, in the way we represent the structure, there are these little sticks. The sticks represent chemical bonds. Now, notice that there are chemical bonds between the sugars and the phosphates, chemical bonds between the sugars and the bases, but there are no chemical bonds between the individual nucleotide bases. Now, this has a really powerful implication for their self-organizational model, because if there's no chemical interaction here, you can't invoke chemistry to explain the sequencing. You can't even invoke the attachment site here, because this bond is called an N-glycosidic bond for people who are keeping track on their scorecards. This bond is the same in every case, so that at each site along the DNA chain, any one of the four bases can attach. It's chemically indeterminate. Any one of those bases can accept, or any one of those sites can accept any one of the bases. The chemistry, in other words, is not determining what attaches or the arrangement of the bases that attach to the medium. The message is here, the chemical message is here, the medium is here, but the chemistry is not determining the sequencing. When Kenyon realized this, he abandoned his whole self-organizational approach. I have a visual illustration, especially for people who aren't chemists. Forgive me. In case the structural formula of DNA didn't make a lot of sense. I've got a little message up here that combines some scientific terminology with American slang. The double helix rocks. All my students used to use rocks as a verb to say they liked something. Now, what we have here is a physical analogy to what's going on in the DNA molecule. In the DNA molecule, you have the backbone, the sugar phosphate backbone. It's not the message, but the medium upon which the instructions are inscribed. I have a medium as well, a backboard, a chemical or a magnetic chalkboard. The little tiny message I have here is also stuck to the backbone with a force of attraction. It's a magnetic force of attraction. But notice that that magnetic force of attraction does not determine the arrangement of the characters in this little informational message. That's determined, well, Michael Polanyi, who first recognized this fact of DNA, said that the information was extrinsic to the physics and chemistry. It's coming from outside the physics and chemistry. Note, we can tell that the magnetism is not responsible for the information because I can quickly rearrange things. We have all the same magnetic forces in play, but we have destroyed the specified information that was there originally. So this is what this helps, I think, understand what's going on in DNA. It's not the physics and chemistry that's determining the messaging. Moreover, if it were the physics and chemistry, it turns out that what you would end up with is not an information-rich sequence, but instead something that information theorists refer to as redundancy, or simple repetitive order. If every time in that DNA molecule you got a T, you turned up a G along the spine, then what you'd end up with is a kind of repeating messages of TG, 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 TG. You'd get a molecular mantra, not a message, not an information-rich sequence or that could direct protein synthesis. So both for empirical reasons and in principle theoretical reasons, scientists working on this self-organizational approach abandoned it. If it had been the case that self-organizing properties were responsible for the information in DNA, we would have been actually incorrect that it was informational. It would have been redundant order rather than specified complexity. Well, all of this got me thinking as I was following the developments of these various failed attempts to account for the origin of information. I had encountered some scientists in the mid-'80s who were entertaining the idea that the information in the DNA molecule might have resulted from an intelligent cause. They were thinking intuitively, and you might just think of that magnetic chalkboard illustration. Where did the information come from? Well, it came from a quasi, at least, intelligent designer, namely me. And there were scientists who were beginning to wonder, one in particular, a man named Charles Thaxton, who wrote a book called The Mystery of Life's Origin, was beginning to wonder if perhaps the information we were seeing in DNA wasn't pointing to what he called an intelligent cause, actual design, not apparent design. And I was aware of these ideas, not entirely convinced at first, and I began to wonder as I got deeper into the question of the origin of life in my studies at Cambridge, was it possible that this design hypothesis could be formulated as a rigorous scientific hypothesis, as a rigorous scientific argument, as an alternative to chance, necessity, or the combination of the two? And oddly, ironically, I became more intrigued with this idea as I began to study the works of Charles Darwin. I was looking particularly at the method of reasoning that Darwin used. And he used a method of reasoning and scientific testing that was called inference to the best explanation. He defended himself against the physicists who said, your theory isn't scientific because it makes no predictions. And he said, well, I make some predictions, but mainly I explain the data, and I hold my theory because it explains the data better than the competing hypotheses. Now, my last year as a PhD student, there was a young, at that time, lecturer coming named Peter Lipton. He later rose to become the department chair of the History and Philosophy of the Science department at Cambridge. He tragically died just a couple years ago of a heart attack, prematurely, in fact. But Lipton helped me a great deal because he was also working on this question of scientific methodology. And he had explicated in some detail this idea of inferring to the best explanation. He said what scientists often do, especially if they're looking at unobservable things in physics or unobservable past events, is they posit that explanation which, if true, would best explain the data. So if you're trying to explain an event in the remote past, you would posit all those different causes that might explain that event, and then you would infer that one which explained it best. Now that raised a deep question in the philosophy of science, which is, what does it mean to be a best explanation? Lipton argued persuasively that in most contexts of science, the best explanation is going to be a cause of some kind. But which cause? Well, it turned out, interestingly, that Darwin and his scientific mentor had worked out some very practical criteria for answering this question. And I first came across it reading the front piece of Charles Lyell's famous Principles of Geology. It had a long, boring Victorian subtitle, but I'm glad I read to the end because it caused a light bulb to go on for me. Principles of Geology, he said, being an attempt to explain the former changes of the Earth's surface by reference to causes now in operation. And when I read the phrase, causes now in operation, it helped me, first of all, to see what the criterion was for deciding what a best historical scientific explanation is. What Lyell was saying here was that if we're going to explain an event in the remote past, we should posit a cause which is known from our present experience to have the capacity to produce the effect in question. For example, I hail from Washington State in the United States. We had a big volcano blow up in 1980. If you go to eastern Washington today, there's a layer of ash that is still discernible across the Palouse country. A geologist finding that, wanting to know what caused it, who may not have been around in 1980, might reason in the following way. might generate, according to Lyell's principles, he might generate a series of possible causal explanations. Maybe it was an earthquake, maybe it was a flood, maybe it was an eruption. But he would then infer that causal explanation, which, based on our uniform and repeated experience of cause and effect, based on our knowledge of causes now in operation, we know is capable of producing the effect in question. In this case, obviously, the geologist would infer a volcanic eruption because we know that volcanoes can produce ash, whereas neither floods nor earthquakes have been witnessed to do so. This was another way of expressing his uniformitarian idea that the present is the key to the past, where specifically now we're talking about our present knowledge of cause and effect being a guide to inferring what happened in the past. Now this, I said, caused a light bulb to go on for me. First of all, it helped me see how the scientists had in very practical ways answered this philosophical question of what constitutes the best explanation. The second thing it helped me do, though, was answer the question I had in the back of my mind about whether or not it was possible to formulate a positive, affirmative scientific argument for intelligent design. About the same time I was reading Darwin and Lyell on this, what is also known as the vera causa principle, invoking true causes, causes known to produce the effect in question, I came across this passage in an information scientist who was a pioneer in the application of information concepts to molecular biology. His name was Henry Quasler, and he said this. He said that the creation of new information is habitually associated with conscious activity. And that really caused a light to go on. Because first of all, I thought, is that true? Well, if you think about that, from our uniform and repeated experience, the basis of all scientific reasoning, according to Lyell and Darwin, it is true. Whether we're thinking of hieroglyphic inscriptions, or paragraphs, or headlines in a newspaper, or sections of computer code, or even information embedded in a radio signal, when we trace information back to its source, invariably we come to a mind, not an undirected material process. So the presence of information at the foundation of life seemed to me to provide a compelling indicator of a prior intelligent cause. One that could be arrived at, the awareness of which could be arrived at, by applying this Darwinian and Lyellian method of allowing our present knowledge of cause and effect to dictate our understanding of what the best explanation of an event in the remote past was. I have the slide here with a quote from Bill Gates. DNA is like a computer program, but far more advanced than any we've ever created. I would argue it's not just like it in respect to its specified complexity, but DNA has the same property of specified complexity the computer codes do. Well, let's think about this from our ordinary experience, from our uniform and repeated experience. What does it take to generate digital code in a computational environment? Well, clearly not wind and erosion, or random number generators, not chance or necessity, or even the combination of the two. Whatever you might think of Windows, they are intelligent beings there at Microsoft that are producing the information. So we have a wealth of experience of agency producing information. Now, in Signature in the Cell, I developed these intuitions in a lot more detail. But I developed the case for intelligent design very consciously using the method of scientific reasoning that I learned about from Darwin, from Lyell, from Peter Lipton, and it has a couple of names: the method of multiple competing hypotheses and the method of inferring to the best explanation. And I look at the attempts to explain the origin of information based on chance, based on necessity, based on the combination of the two. And I infer that intelligent design provides the best explanation because each of these other forms of explanation have been shown for various reasons, either empirical, theoretical, or both, to be incapable to produce the information necessary to produce the first life. Now, coming back full circle to where I started the lecture, as we think about Darwin's legacy, my own view is that his enduring legacy is not that he refuted the design argument, but instead that he established the legitimacy of questions about design, real, whether it be real or apparent. That's a legitimate scientific question. He answered it one way. I'm now answering it another way. But that he also used and formulated a powerful method of scientific investigation that allows us to look at these questions of origin, to look at life's history as a properly scientific endeavor. And I have used that very method to make my case for intelligent design. Now, one final postscript. I have made the case for intelligent design tonight, though not in the book, relying only on what you might call molecular biology 101, the very basic things that have been known since the late 1960s. It's striking that these undirected materialistic theories of evolution, of chemical evolution, have been unable to account for even that. But in recent years, especially since the publication of the results of something called the ENCODE project, we have been learning a lot more about the information-bearing properties of DNA, the informational properties of the whole information processing system in the cell. And I had a conversation recently with one of the programmers that works closely with us at our lab. He's an elite architect-level programmer at Microsoft. And he walked into my office one day and threw a book down on my table. It was called Design Patterns. It's a standard design manual for software engineers. And he said somewhat dramatically, especially for a software guy, he says, "I get the eerie feeling that someone figured this out before us." And I said, "What do you mean?" And he said, "Well, you know, Meyer, you're always going on about all the digital code in the DNA." And I said, "That is impressive. I get that." But he said, "What impresses me more is the design patterns, the design strategies that are clearly at work in the information processing system in the whole of the cellular gene expression system." He'd been doing programming to represent that in a simulation. For one thing, we know that the junk DNA, what we used to think of as junk, the non-coding regions of the genome, are now known to be functional, and overall this junk DNA, which is no longer junk, the non-coding DNA, is functioning like an operating system, which is directing the timing and regulation of the data files, the DNA coding regions of, the protein coding regions of the DNA. There's automated error correction. There is a files within folders hierarchical filing system, both in the cell and in our complex digital computing world. There is a mode of digital storage and retrieval of informational modules. The cell needs a certain kind of protein to be built. Oftentimes it will go and find different data sets stored in different places, access them, concatenate them, and build a completely new protein on a needs basis. My computational friend, my computer engineering friend, was saying, this is mind-boggling because we use all these same design patterns, these same design strategies. And then he also talked about the nested coding of information, what's called dual coding or dual encryption. So the case for intelligent design is based on the molecular biology 101, but I think it's gotten even stronger because of these other things we're learning about the information processing system of life, and how that system manifests features which, again, are produced by one and only one known type of cause, that being intelligence. And so, for all those reasons, I've concluded that it is possible to formulate a scientific theory of intelligent design, and in fact, at present, that theory, I would argue, provides the best explanation of these crucial informational features of living systems. Thank you very much.
Time is just about up. I think we must have one, and I happened to notice table 6 first, so...
Basically, what is science?
Thank you.
Now, the reason I'm asking this is that you've regularly been saying this is a scientific argument, but we have academic institutions and professional institutions around the world coming out saying intelligent design is not science.
Right.
And
I've heard that before, actually. Yeah.
So there's something wrong here. We need some clarification about what is science.
Well, this is where, by the way, I have a colleague here, Douglas Axe, who's a protein chemist and molecular biologist, and he directs our lab. My background is in the philosophy of science. And it is the discipline of philosophy of science that addresses these questions of the definition of science. It turns out defining science is a notoriously difficult thing to do. It's a semantic debate. It's a debate about definitions. And depending on how you define science, you can either exclude or include within the designation of science all manner of different theories and competing ideas. Darwin faced it, interestingly enough, in the 19th century. Herschel and others called his theory of natural selection by random variation the law of higgledy-piggledy. And they caricatured it as a non-scientific thing because it was not based on a law-like process. It was strictly law-like. And that's where he invoked inference to the best explanation to defend himself. My case, fundamentally, I don't care whether intelligent design is considered to be science or not. That is not the most important question. That is a semantic question. The most important question is whether it is true, or whether it's likely to be true, or most likely to be true, given the evidence we have. What people have done to avoid answering that more fundamental question is repair to these semantic arguments. Oh, it's not true because it's not, intelligent design is not science, therefore we don't have to consider the case for it. I don't think that follows. Whatever you call it, there is a compelling evidential argument for it. But I also think that using very basic understandings, a very credible definition of science. Typically people want to demarcate science from non-science by invoking an understanding of, by reference to method. Science is different than non-science by reference to the methods it uses. Well, the case for intelligent design, and this is why I emphasize this in the lecture, is based on the very same method of reasoning that Darwin employed. So if you want to argue that intelligent design is not science, then applying that same criterion, that same yardstick, you will have to exclude Darwinism from consideration as a scientific theory. Now, I happen to think both are scientific theories. I happen to think chemical evolutionary theory, the actual opposite number of my case for intelligent design, is a scientific theory. The definitional question should not be dispositive. That's just a matter of how you categorize an idea. That's not fundamentally interesting. What's fundamentally interesting is whether it's true or not. But the way people do this categorization is they're called demarcation arguments. And they're notorious in philosophy of science because they always fail. And let me illustrate. One of the simple demarcation criteria that's been offered is the idea of observability. If something's going to be a scientific theory, if a theory is scientific, it must not invoke unobservable entities. Well, ask Professor Josephson in theoretical physics about whether there are unobservable entities that ought to be part of science. Or consider Darwinism. Darwinism invokes unobservable past events, transitional intermediate forms that are not observable, and yet or past mutational events, not observable. If you make observability, a strict criterion of a theory being scientific, we have to disqualify all kinds of theories that are considered scientific, including intelligent design. And so what I found is as you examine these demarcation criteria, what I found is that if you apply them very strictly, then not only intelligent design, but all competing evolutionary ideas are disqualified from consideration as science. If you apply them more realistically, taking into account the nature of the inquiry, that it's an historical scientific inquiry, then both intelligent design and competing evolutionary theories are properly considered to be scientific. So in short, I think the objection is a red herring, but if you take it seriously and let people define what they think science is, invariably it does not do the work of settling the debate, of discriminating between one approach and another. So the biggest example of this was the federal judge in the United States in the Dover trial who wanted to disqualify intelligent design. He said it wasn't science. And I was asked in the media my thoughts on this, and I said, well, we actually don't look to federal judges to settle deep and imponderable questions about the nature and definition of science. It's a different discipline is in charge of that. And so anyway, that's my not so short answer.
Well, thank you very much. Unfortunately, the time is past that has been allocated for this magnificent occasion. And I think we're all extremely grateful to Stephen for his very clear explanation. As I understand it, the basic point is that in the cells there is a fairly substantial program which works in producing the life as an arrangement of the amino acids. The arrangements are such that mere chance or a mixture of chance and necessity wouldn't demonstrate the way in which such a working system or organization could evolve or come into being. If we look around the world, the only way in which working systems of that kind have been put in place is as a result of the operation of human minds. In other words, intelligence. And therefore, if you look at what the possibilities are, the only possibility that at the moment seems to work is the possibility of an intelligence operating to produce that sequence. And I think the stance of Stephen is that at the moment, this is the best available theory. If in time some better theory comes, well, it would displace it. But until that happens, Stephen's hypothesis is that this is the best explanation of that fundamental aspect of human life. I had the privilege once of asking Richard Dawkins what he meant by life. I'm still waiting for the answer. Well, thank you very much indeed, Stephen, and your colleague, for being here with us tonight, and for all you've done in the pursuit of science in this area. And I think we also wish to thank very much those who have provided this occasion for us as an opportunity to hear for ourselves the arguments that Stephen has deployed in his book and explained verbally, which for some of us is easier to follow than reading. Otherwise, for some others, some find it easier to read than to listen. But we've had the privilege of having the opportunity to do both. And for that, we're very grateful. And thank you very much to the institution and to the particular people who so kindly provided this, and to all those in the premises who have served us so well with such a delicious meal and within the sort of confines and restrictions that are involved in mixing science with food. Thank you very much indeed.
Thank you for your good summary. I should have had you do the lecture.
Thank you.